Welcome everyone. Thanks for joining us online today for the class. I apologize that I'm not able to make it to campus on Wednesday, but hopefully you're watching this in the comfort of wherever you like to take in genetics. Um, so for today's lecture, we're going to just finish up a few things on Chapter 6. Obviously, those were not covered in the last exam. Um, and then we're going to start on Chapter 7. So for Chapter 7, you might notice as we're going through the material for today, some of it you may not have read in the first 8 pages or so of Chapter 7. The reason for that is the book breaks up the Chapter 7 material by the first half being the types of mutations and the second half being the ways that they're fixed. I like to present the material together, addressing individual types of mutations and how they're fixed at the same time. So what I'd really like people to do is make sure that by Friday you've read through chapter or through page 150 in chapter 7, and you'll have then covered all of the information that we'll address by the end of the lecture on Friday. So for Friday, just make sure that you've gone uh, through chapter 7 through page 150. So, like I mentioned, today we're going to just finish up talking about translation. We're going to talk about the different steps of translation that we really haven't gotten into much detail yet. We're going to go into very little detail when it comes to these steps. The book goes into a lot of detail. I think that is more for a molecular biology class, and I think your time would be better used focusing on some other aspects of genetics. So we're just going to touch upon the most important parts of these 3 stages of translation. That's all you're going to be responsible for. And then we're just going to talk briefly about the difference between prokaryotic and eukaryotic translation, and particularly how this plays a role in disease. And then in chapter 7, we're going to start talking more specifically about mutations, the types of mutations, how they occur, how they're fixed. So the first 2 lectures in chapter 7 will be based upon, mostly based upon what's in the textbook. And then the 3rd lecture in chapter 7, we're going to be using a primary literature article to talk about an interesting type of mutations that has to do with what are called transposons. So we can break translation up into 3 steps. Just like we actually were able to break up transcription into 3 steps, we can break translation up into 3 steps. The first step of translation is initiation. So when the pre-mRNA is transcribed in the nucleus by the RNA polymerase, it has to be, and when the RNA is transcribed in prokaryotes, that RNA can be translated immediately, because remember, prokaryotes do not have nuclei, so translation can start right away. In eukaryotes, that pre-mRNA is transcribed in the nucleus. It gets modified, so there's a 5' cap, a 3' tail, and introns are removed, and that gets exported to the cytoplasm where that's able to be translated. When we talk about these steps, these are essentially the same in prokaryotes and eukaryotes until we talk about the specific differences in a few minutes. So in both of these types of organisms, the first step of translation is initiation. Initiation occurs when the mRNA, so this is our mRNA sequence right here, 5' to 3', the mRNA is bound by what's called the small subunit of the ribosome. So we can break the ribosome up into 2 subunits, a small subunit and a large subunit, and each one of these subunits are made up of a number of RNAs and a number of proteins. This small subunit binds to the mRNA at what's called the mRNA binding site. And you can see here, the small subunit binds at the mRNA binding site, and this is a large protein complex, so it also is binding at the same time to the start codon. Because remember, in eukaryotes, there are large untranslated regions of in the RNA. In prokaryotes, there are regions that are not translated, short regions that the small ribosomal subunit binds to. So the small ribosomal subunit binds to this mRNA, and this recruits the first tRNA. This is called the initiator tRNA. And this is always going to carry methionine, because remember the first codon is always AUG. And when this occurs, guanine triphosphate, GTP, is hydrolyzed. So one phosphate bond is broken here. GTP is converted to GDP, and this energy that is released is what allows initiation to begin. And this energy release helps recruit the large ribosomal subunit. And at this point, this tRNA is in what's called the P site. So on the bottom here, this kind of sums up initiation. So the small ribosomal subunit binds the mRNA, recruits the first tRNA, and then this results in the recruitment of the large ribosomal subunit. Elongation is a cycle because the elongation is when every single amino acid is being added. In elongation, a new tRNA arrives at the A site. More GTP is hydrolyzed. When this new tRNA is at the A site, the growing amino acid chain is transferred from the tRNA in the P site to the A site, and this is where the peptide bond formation occurs. And this is one of the important things that I want you to understand about elongation. This is where these peptide bonds are formed. These bonds that are present in between the individual amino acids in the polypeptide chain. And this occurs by the enzyme peptidyltransferase. So you can see the amino acid chain is moved over from the tRNA in the P site to the one in the A site. So we're now here. And then what happens is the tRNA translocates. It shifts over from the A site to the P site. This tRNA that previously had the amino acid chain gets ejected, and the cycle continues. So the most important thing that's happening here is that there's this peptide bond that's being formed. So amino acids are added one by one to the preceding amino acid. Termination is obviously the last step. In the last step, termination is signaled by the presence of a stop codon. So we know that there are three stop codons, UAG, UAA, and UGA. And stop codons signal for a release factor to come to bind to the A site. When the release factor binds to the A site, this cleaves the polypeptide from the tRNA that's in the P site and disassembles the ribosome. So this release factor is what signals the ribosome to cut that polypeptide, which is now a free polypeptide, and can go and do the work in the cell, and also signals for the ribosome to disassemble. This is the amount of detail that you need to know about translation. The book goes into a lot more detail. That it's not necessary. What I went over is all that you need to know. There are a couple videos here that you're welcome to watch. This is a very classic video, and I apologize if there's sound in the background. They seem to be mowing my lawn.
Only rarely is there an opportunity to participate in a molecular happening.
So this is a very well-known classic video that was filmed in the 1960s at University of California, Berkeley, where a number of people, many undergraduates and graduate students, took part in a reenactment, in a way, of protein synthesis. And you obviously don't need to know this for any exam or anything like that.
You're going to have that opportunity.
But it's interesting.
Translating the message. First, the small subunit with the aid of an initiation.
Fly the 30S ribosome did gyre and gimbal in the wave. Gathered in tRNA and came galumphing back.
So if you have a chance, just take a look at it. It's really quite amusing. There's another video here. It's a much shorter video. This is a video that sums up translation. It's about 3.5 minutes long. I'm not going to play it here because obviously you can play it on your own. The URLs are shown here on the slide, so you can copy those into your browser. But again, when we're talking about the steps of translation, this is the amount of detail that you need to go into. Your book goes into a lot more detail. I'm not asking you to know that much, but you should know the role of the small ribosomal subunit, the large ribosomal subunit, these where the tRNAs bind, where the peptide bond formation occurs, so on. Obviously, I can't take any questions when I'm giving this this lecture online, but if you have questions after viewing this lecture, please come find me either after class, during office hours, or any other time. That way, make sure that you have a good idea of what's going on here. And just a reminder, there are these animations that come from your book. These are posted on eClass. These are also helpful in understanding the steps of it says initiation up here, but these are animations for initiation, elongation, and translation. So please do make use of these. So I mentioned that the small subunit of the ribosome binds to this mRNA binding site, which is upstream of the start codon. So you can see it's before the start codon on the 5' side. In bacteria, this is what's called the Shine-Dalgarno sequence. So it's about 5 to 8 nucleotides upstream of the start codon, or the initiation codon. And the way that this works is the 16S ribosomal RNA base pairs with this sequence. And that's how the ribosome knows where to start translating. So there may be multiple AUGs in a particular coding sequence. The presence of the Shine-Dalgarno sequence is what tells the ribosome this is the AUG that we're supposed to start translating at. And as is the case with every time that nucleic acids form hydrogen bonds with each other, you can see that it is anti-parallel. So this is the mRNA in the 5' to 3' direction here. The 16S ribosomal RNA is anti-parallel, 5' to 3'. So this is an RNA that is part of the small ribosomal subunit. So this RNA reaches out from the small ribosomal subunit and finds that Shine-Dalgarno sequence, and that tells the ribosome start translating at the AUG that is just downstream of that right here. So this is kind of a mock-up of, imagine you have this small subunit of the ribosome with this 16S ribosomal RNA sticking out from it. And this makes sure that the ribosome reads the mRNA in the correct spot, and it also makes sure that it's in frame. So remember, we talked about frame shift mutations. Codons read every 3. If the translation doesn't start at the right nucleotide, if it starts off by 1 nucleotide, so instead of instead of starting at A here, it starts at U, or it starts at this U, everything's going to be off by 1, and all of the codons are going to be wrong. So this is important for making sure that the mRNA that's being read is in frame. Now, think for a second, what do you think would occur if there was a mutation in this Shine-Dalgarno sequence? Obviously, you can't click in, but just take a minute, think it over. So most likely, translation would not occur, the initiation of it, because there would be no way for the ribosome to know where to bind. And if it doesn't know where to bind, it doesn't know where to start translation. Now, it's possible that C could occur. Translation initiation could occur at the wrong location if there was another sequence that was very similar, or actually identical, to the Shine-Dalgarno sequence. So B is the most likely result, in that initiation of translation would not occur. But it's possible that C could occur if there is a different Shine-Dalgarno sequence, but that's rare that that would occur. Now, it asks down here, what if there was a mutation in the Kozak sequence? Well, in order to answer that question, you need to know what a Kozak sequence is. And if you remember from your reading, the Kozak sequence is essentially the eukaryotic version of the Shine-Dalgarno sequence. It is the location in the mRNA that the eukaryotic ribosome binds to initiate translation. So if there was a mutation in the Kozak sequence, you would have the same effect, except this would apply in eukaryotes instead of prokaryotes. Now, while I don't want you to necessarily understand all the intricacies of the steps of translation, it is important to understand the differences between prokaryotic and eukaryotic translation, because this is very critical when it comes to, say, the treatment of diseases and the causes of diseases. So we've talked a little bit about differences between prokaryotic and eukaryotic cells. We talked about that in the first lecture. And there are a number of differences. One of those is very cogent to translation. So in bacteria, the ribosomes are smaller than they are in eukaryotic cells. And you can see here it says ribosomes are small, called 70S. Ribosomes are large, called 80S. We'll talk about what that means in just a second. But the understanding of these different ribosomes is actually relatively new in a lot of ways. Ribosomes were discovered back in 1955, but it's taken a long time to truly understand what's going on with them, to the point where the Nobel Prize in Chemistry in 2009 was awarded for the studies for the structure and function of the ribosome to these three fantastic scientists right here. So it's still a field of study that's very important and ongoing. So the difference between ribosomes in bacteria versus eukaryotes really is just the size. The function works the same, and the different subunits actually look very similar. They're just different in size. And S here, when it says 50S, for example, S just stands for what's called Svedberg units. It's just a way to measure size by centrifugation. You don't need to know what that means, but you should see that here that in prokaryotes you have this 50S subunit and this 20S and this 30S subunit, and in eukaryotes you have a 60S and a 40S subunit. And then there are these other ribosomal RNAs that differ in size as well, and the number of proteins are slightly different. But these ribosomes, the large subunit and the small subunit in each of these types of organisms are different. Both prokaryotes and eukaryotes use the same tRNAs because we know that the genetic code is universal. And we know also that ribosomes are made up of RNA. So the RNAs that are made that populate the ribosomes are transcribed by polymerase I and polymerase III in eukaryotes. And ribosomes are also made of proteins. Those are transcribed by polymerase II in eukaryotes. Prokaryotes, there's only one polymerase, so they're all transcribed by the same polymerase. So based upon that, let's think about what is the importance of this difference. So let's say you're a healthcare provider and you come across a patient with a really bad bacterial infection. So as we normally do, we treat those with drugs, typically antibiotics. But the pharmacy forgot to put the name of the drug on the label. Bad pharmacist. Now, let's say you have a description though of what each drug does. So drug A inhibits the peptidyltransferase activity. So this is the enzyme that forms the peptide bonds during elongation of the 60S ribosomal subunit. You have drug B, which inhibits the peptidyltransferase activity of the 50S ribosomal subunit. And you have drug C, which causes premature termination of translation by mimicking tRNAs. And what that means is this drug looks like a tRNA, but when it's used during translation, it stops protein synthesis instead of adding on a new amino acid. So which of these would you want to treat your patient with? Think about it for a second. Well, these are actually examples of antibiotics that are currently available and are used for a number of different things. Now, if you picked, well, before we get to which you picked, based upon this information, you could determine that this one drug, cyclohexamide, up top here, this is going to block translation in eukaryotic cells because eukaryotes have this 60S ribosomal subunit. Chloramphenicol blocks translation in prokaryotic cells because it has this activity against the 50S ribosomal subunit. And pyromycin, this one down here, this is going to block translation in both because remember, prokaryotes and eukaryotes both use the same tRNAs when it comes to translation. So, in fact, the answer was B, the second one, drug B, because this would only block translation in prokaryotic cells. Cyclohexamide you would never really want to use as a way to treat a bacterial infection, but it's used oftentimes in research to try to better understand the different processes that are involved in gene expression. There are examples of diseases that occur due to inhibition of translation in eukaryotes. So a good example of this is actually the disease diphtheria. So diphtheria is very rare nowadays, but back many years ago in the 30s, 40s, even into the 50s, diphtheria was relatively common until a vaccine came about. Now, there's an organism called C. diphtheriae, a bacteria, and this produces what's called diphtheria toxin. So a lot of these bacteria can produce toxins, and this toxin, what it does is it blocks this EF2, elongation factor 2, protein's activity. So it blocks the ability of the tRNA to bind to the A site in the ribosome. So this is actually how diphtheria is deadly, especially deadly in children. This diphtheria toxin blocks this EF2 protein, and the incoming tRNA can't bind to the A site, a peptide bond can't be formed. And this is the reason that you should get vaccinated for things like diphtheria, because it's a terrible, terrible disease, and vaccinations prevent the proliferation of this bacteria that can release this toxin and specifically kill eukaryotic cells. So that's the end of chapter 6. Please let me know if you have any questions on chapter 6. This part of chapter 6 will be fair game for the next exam. So there's plenty of time before the next exam to deal with any questions that you have about any of this material. We're going to move on now to chapter 7. So chapter 7, we're going to talk about DNA mutation, so different types of mutations in DNA. We're going to talk about how those are repaired. And finally, we're going to talk about these really, really interesting things called transposable elements, transposons, and how they can cause mutations and how they give us information about different things when it comes to the genome. So it's probably helpful to start out by defining what a mutation is. We use this word all the time. We use the word mutant, we use the word mutation, but we probably need a definition for this. Your book has a definition. The definition is the process by which the sequence of base pairs in a DNA molecule is altered.